Hereford, and here is my light reaction and Calvin cycle diagrams. So we're gonna start with the light reaction and we'll get into the Calvin cycle. So this is a basic breakdown of the light reaction. This is the granum, and this top is the thylakoid membrane and thylakoid space, so that little tiny part. So in the membrane of the thylakoids, you have photons of light coming in, hitting chlorophyll A, B, which is the site within photosystems, and then those photons bounce off and hit reaction center, chlorophyll A. And then we have an electron transfer to a primary electron acceptor. Enzyme uses water for transferring electron to oxidize chlorophyll and produce O2. And so here's my basic diagram for that. I'll go really slow and let you look at it for a minute. Okay, so this is the lumen, which is inside the thylakoid membrane, and this is the stroma, which is outside the thylakoid membrane. And we have photosystem 1 and photosystem 2 here. And you see there's, enzymes coming in and going out, and some enzymes come down here and some go through here. And so right here, and you see the light coming in. So we have the plastoquinone, and this is just involved in electron transport. And then we have the B6F complex, and this is in the cytochromes, and these are capable of performing electron transfers and catalyzing by either reduction or oxidation. And then as you continue through, well, let's go back down here. So we have the plastocyanin, and this is another, it's an electron transport process associated with the whole idea of photosynthesis. So as we have an electron, make its way up here, it goes to the ferredoxin, which is a protein that, again, mediates electron transfer. And then the ferredoxin NADPH reductase is the last enzyme that does electron transfer in photosystem 1 before it turns into NADPH. And this part should really be up here, except I didn't have enough room. So you'll see that NADPH and ATP go into this Calvin cycle, but we'll get to that in a minute. So we have hydrogen ions going in right here, and we have our ADP plus our phosphorus, and then so we're making ATP. And this in the ATP synthase. So we have ATP and NADPH in the Calvin cycle. So we go into the stroma, which is outside of the thylakoid membrane, and this is anabolic. So in phase 1, Rubisco joins RUBP plus CO2, and that turns into 2 molecules of 3-phosphoglycerate, and Rubisco is just an enzyme. It helps with carbon dioxide and oxidation. So then we go to phase 2, where reduction happens, and we have ATP plus 3-phosphoglycerate, and then we have phosphorylation happening, and then we have 3-phosphoglycerate plus ADP, and this has reduction by NADPH. Then from here, we go to the glycerol 3 aldehyde, or G3P plus phosphorus, and we get glucose, or regeneration of RUBP. Thank you. Bye-bye.